This is your chapter 15 notes on genetics. It talks about uh, more complex methods of inheritance, just besides Mendelian, more chromosomal. I'm just going to be covering 15.1 to 15 sections of point 1 and 2 for this first video. Okay, remember that genes, which are just sequences of DNA, are located on chromosomes. So here's a chromosome. Here's a complementary and homologous chromosome, one from mom, one from dad. And genes are just right all over those chromosomes. Matter of fact, there are lots of genes on individual chromosomes. The locus is the location of where the gene is located on a chromosome. I'm going to show you a handout that shows some of the all your 22 pairs of chromosomes plus your sex chromosomes, what genes scientists have already identified in their locus, what chromosomes are located on and where on that chromosome they're located. So chromosomes are just a bunch of coiled up DNA. So if you take that out and you stretch it, it's going to be that double helix structure of DNA. And then if you get really close and look at it, it's going to contain the sequences of nitrogen bases, the A, T, G, and C, which is what all of your DNA is made out of. And those sequences, what order those bases are in, are considered your genes. The reason genes are different in different people is because that sequence can be slightly different. Figure 15.2 is a good summary of Mendel's law of inheritance and how they relate to meiosis. I encourage you to go back and kind of review that since it's been a while since we talked about meiosis and how alleles are inherited, dominant, recessive, through independent assortment and random fertilization. I encourage you to go back and look at that. Or I could show it to you now. Good, I should have looked at my PowerPoint ahead of time so I knew what I was going to show you. So this goes through and talks about how that happens with meiosis. How do we go from all these genes? Now we've added in the alleles that are going to code for different traits on the chromosomes and showing how we produce gametes, and those gametes come together through fertilization. So if this was the parental generation here, this produced their egg and sperm, and then those would come together, and then from there, these organisms, this is passed on a big R, big Y, little r, little y. So they have both of these, and then these are going to undergo meiosis, and they're going to produce their own gametes that then can make the next generation. The F2 generation is a combination of mating the two of the F1s. So this looks at actually how do we get those alleles? They come from our egg and our sperm, and how are they assorted? Well, they're assorted randomly depending on, in that metaphase plate, how they line up. Notice this side, the big R and the big Y are on this side, so they go into these cells, and the little R and the little Y are on these sides, so they go into these cells. But in this is flipped right here. And so the little R and the little Y are going to go together, and then the big R and the big Y, which is why you a lot of times will see blue eyes with blonde hair, but not always. You can see brown eyes and blonde hair. Now, those are kind of an example of just how this works, but honestly, blue eyes and blonde hair and eye color in general and hair color are not very simple, big R, big Y, little Y kind of genes. These are actually a combination of many genes put together. But you get the idea of how not necessarily one gene is always going to go with the other. Those alleles separate independently, and that yields different egg and different sperm. So let's talk a little about sex-linked traits. Thomas and Morgan experimented with fruit flies and found that some inheritance patterns that were not like Mendel, so they didn't seem to follow that same dominant recessive pattern. The most common type of eye color is red, which is what we call the wild type. White eyes are actually a mutant type, and it showed up in different ratios in males and females. And this is where he was like, this isn't like Mendel's. Mendel's, it shouldn't matter what gender they are. You can read more about his experiment and how he determined that eye color was carried on X chromosomes on these pages. Especially if you're interested in history, it's kind of important to look at that and see, like, how did he come up with this idea? But ultimately, what he determined based on the genetics and the ratios that he got is that the eye color gene is located on the X chromosome. And because of that, females are XX and males are XY. And so for females, they can have 3 different options of genotype, which means that these guys will be one color and these will be different. But males only carry 1 copy, so they only have 2 options, but it's not going to be the same. They can't ever get this heterozygous form. So here's the eye color. We're going to be looking at some fruit flies, and one of the traits we're going to be looking at has to do with our eye color. In humans, XX or XY determines sex, which is typical of most mammals, but varies in other species. In humans, the Y chromosome has the SRY gene, but not much else. Y-linked genes are on the Y chromosome, and there's not many of those. And X-linked genes are on the X chromosome, and there are many of these. The Y chromosome is not very big. That's partly why there's not a lot of genes on there. But there are a lot of genes on the X chromosome, which means that there are a lot of traits that are sex-linked. They're linked to your gender, and there's a different number of males that have it and different number of females because males only carry 1 copy of the X and females carry 2 copies. So this just shows different systems, like in humans it's the XY system, so XX are females, XY are males. This is a cockroach. They have an X0 system. So males just have 1 X, they don't have anything else, and then females have 2 X's. This type of bird, not really sure what that is, has a ZW system, and so their males are the homozygous, they have the same, whereas females do here. And then the heterozygous is actually a female. And then this looks like an ant. They have a haplodiploid, and so they don't really create, I guess, egg and sperm per se. They just have something that is a haploid cell and something that is a diploid cell. They don't have, and they may create egg and sperm. I'm really not sure about ants, but what's important here is that their offspring or their zygotes, it depends on how many chromosomes their entire cells have. That's kind of interesting. I wasn't aware of that. X-linked traits. They are expressed differently in males and females because females have 2 X chromosomes and males only have 1. If an X-linked trait is recessive, that means that males can't be heterozygous. They can only be this or this, and of course that should be written subscript. It should look more like this. Superscript, not subscript. If the recessive form is defective and leads to a disorder, males only need 1 copy of the gene to be affected. That's really important. That means more X-linked disorders are found in males. Okay, the general idea here. So normally a female, say this is a recessive disorder here, a female would have to have 2 copies in order to be affected, but a male only has to have 1 copy because he doesn't have the other backup good copy like a female. You're going to find a lot of females in recessive disorders are heterozygous, and they appear normal because they've got the normal version of the gene. Fathers pass X-linked genes onto their daughters, but none of their sons. You can spot that pattern in a pedigree, which we're going to be analyzing some of those. That's kind of a key indicator that it's an X-linked, is because the father will have it, but none of his daughters do. Well, let's look at kind of how that happens. Here's dad, and here's mom. Okay, and so we have a recessive disorder. Dad has it, and mom is, we'll say she's heterozygous. And so if they produce a female like that, they will be a carrier, but if they produce a female like this, they will actually have the disorder. Either way, dad is passing on the X chromosome to him. You think about it. If it's a boy, dad's passing on the Y, right? If it's a girl, he has to pass on the only X he has. And then the boy would be normal here, and then because mom is heterozygous in this case, that, excuse me, that there's a chance that their son would have it because he only has 1 copy. But dad's not passing, that's coming from mom. If mom was normal, then none of the sons would have it at all, but the females would be carriers, passing it on, and then they then can pass it on to their children. And they could pass it on to their sons because they have 2 X's and they can go off up to see what they're passing on there. Males that receive the recessive allele from their mother will express the trait. So, again, say this mom then is going on and marries somebody even that's normal. She would pass on that to at least some of her sons. So here's pedigree showing X-linked disorders. This one's an X-linked dominant, and so that means to be affected, you would have to be this or this the female and this the male. And X-linked recessive means that to be a female, you have to be this only and male this. So you can see X-linked dominant disorder here. In a pedigree, which we haven't really talked much about, if the boxes are shaded, that means they're affected by it. They have whatever it is you're looking at, whether it's a disorder or a trait. Squares are men, circles are females. And then anytime two are combined like this, that means that there's a marriage and they've mated. Well, we assume marriage, not always. They've mated, and then anything branching off of there, those are going to be all of their children. So these four were their children, and then this child married that female, and then these two here had these four children there. So that's how pedigrees work. There's some other symbols in there that if you get complicated where you get twins or you get a marriage in a family, there's some other symbols, but these are the main ones that you would have to know. And this shows generation 1, generation 2, or what information they have or how far they have it. And you can see that if this is a X-linked disorder, this female would have, or sorry, an X-linked dominant disorder, she would have to have a capital B there. And the female would have to have well, she at least has to have one normal B, but we don't really know if the other one's affected. We could figure that out based maybe on our children. So here you have a female that is from dad, and then we don't really know what the other one is from mom. Affected, so probably it could be a little bit though. And then we can even maybe trace that down into the children to see what they have. And then other children here are normal, so that means that it's a male. So mom had to pass on a big B, or had to pass on a little b because this is a dominant disorder. So we know that mom has to be, oh wait, she has to be little b, I didn't use the recessive disorder. So mom has to be this because this is a dominant disorder. Okay, that was my wrong there. So these would indicate affected. Do with their genotype. So unaffected would be little b. So mom had to pass on a little b here. And then same here, little by. And you could go on and you could assign genotypes to these individuals. This guy's a Y, so he actually got, this is a female. Sorry, this is their child here. That's not their child, that's the marriage. This is a female, so it's going to be X. So affected little b. So I know it was affected. So big B from dad, and then little b from mom. And then this is a male, so Y from dad. I'm so used to thinking of being recessive. That is going to be unaffected because it has to be a big B for them to be affected. And then you could keep going on and you can look at the children. So this is a male that does not have it. And then they had a child that is male that does. So mom had to pass on that little b. So, and you can keep going and you can assign genotypes all the way down. This is actually the more common, which is why my mind is automatically thinking recessive, because most X-linked disorders are recessive. So here, we've got a female that is like this. Okay, now remember before that was unaffected. And here it is because this was a dominant and this is a recessive disorder. And then the male is going to be like this because he is unaffected. And then you could start assigning genotypes to their children here. Unaffected, so that means if this is a recessive disorder, they have to have at least one big B. And then the other one, we don't really know yet. You can figure that out later. Big B there. Oh, wait, had to get, if you got big B from there, then little b from here. You know that one. And then, let's see, this child would be affected, so it would be little b and Y. and little b and y, and this one would have to be at least one big B and then one little b because that's the only other option up there. And then x little b and x little b. And then notice that because mom has that and dad has that, and they're both affected, all their children are going to be affected. There's no way around that one. Whereas here, this is a male, must have gotten little b from mom because he's affected, and then a y from dad. And we don't really know, well, we do know because dad is not affected, so he had to have a big B there. So there's a way you can figure out usually the genotypes of almost anybody in the pedigree if you know that pattern of inheritance. Usually, though, you get a pedigree and you have to figure out how it's inherited. You have to know that this would indicate sex length. A couple of sex traits of interest, Duchenne's muscular dystrophy and hemophilia. Read about those in your textbook. Duchenne muscular dystrophy has to deal with the loss of muscle and the ability to basically control your body movements because you don't have muscle tone. The muscle fibers break down, and then hemophilia is a blood clotting disorder, and so they tend to bleed to death. They have to have a lot of blood transfusions. Little bumps and things that if you bumped against the table, it probably wouldn't hurt you, but internal bleeding can happen in somebody that has hemophilia because they don't clot. A small paper cut can be a problem. So read about those in your textbook. Solving X-linked genetics problems. Whoops. Didn't mean that. These are really going to be written the same way and solved the same way. We just write the allele superscript to the chromosomes, and I've been doing that to kind of show you. And we solve the same way we would solve a typical genetics problem. You saw me do the Punnett squares before. There's no difference. The only thing here is that you have to consider the offspring, and that half of these boxes are going to belong to females, and half of them are going to belong to males. Now, why does that matter? Well, it matters because if you're talking about like a question about how all the daughters that they have, well, then you're only looking at 2 boxes. If you're talking about all the possible children, then you're looking at all 4 boxes. So it changes a little bit when you're calculating probability, depending on what the question is actually asking. And we'll practice that. So here's showing the different ways sex length can go about with the egg and the sperm and the different options there. X inactivation can also be referred to as a bar body because females have 2 copies of the X chromosome. Most of one of the X chromosomes is inactivated, so they're not having too much genetic information during early embryonic development. Therefore, the cells of males and females have the same effective dose of most of the X-linked genes. This makes sense because otherwise females would have a lot of extra genetic information, and males would have about half of that. The inactivated X in each cell of female condenses into a bar body. These genes are not expressed. The bar body chromosomes are reactivated in the cells that give rise to eggs, so gametes will have an active X. Which X is inactivated is random and different for each cell during embryonic development at the time of X inactivation. Each cell after that original cell will divide by mitosis, so each subsequent cell will be identical to the previous cell. Females can be mosaics of 2 types of cells. Those with the active X derived from the mother, and the active X derived from the father. This is kind of interesting with a few things here. If the female is heterozygous, this means that about half of her cells will express one allele, and half will express the other. That's kind of cool. The example is a tortoiseshell cat. Notice the patchwork pattern to the tortoiseshell. It's because some of the cells are expressing one allele and some are expressing another allele. In humans, an example of a mutation that affects sweat glands. A woman who is heterozygous for the trait has patches of normal skin and patches lacking sweat glands. This is X inactivation in this form is really much rarer in females. There's not a lot of things that will come out as a mosaic like that, but there are calico cats is another example, tortoiseshell and calico cats that have that thing. Usually, again, all the cells are divided by mitosis, and so it's the same, whichever one's inactivated. As you can see here, black fur, where this is the active X, and then orange fur, where this is the active X. So it's really kind of cool. And you rarely ever find a tortoiseshell that's male because you would have to have both X's in order to get that. And we're going to stop with those here. The other one is, if you read about it or look about this, Calico Cat, same idea.